Hello again, Bio 220 students for online microbiology. This lesson will cover Chapter 14, Principles of Disease and Epidemiology. Some terms that we're going to cover: pathology, which is simply the study of any particular disease; etiology, which is the cause of a disease; pathogenesis, which is how a disease develops over time; infection, which is the invasion or colonization of the body by pathogens; and disease, which is an abnormal state in which the body is not performing normal, typical functions. Some more terms for normal microbiota, or your typical bacteria flora. There is transient microbiota, which may be present for days, weeks, or months. There is normal microbiota, which will permanently colonize the host and does not cause disease under normal conditions. Though if, say, intestinal bacteria reaches parts of the body outside the intestine, that would be a non-normal condition that might cause infection. There is also a Human Microbiome Project, which analyzes the relationships between microbial communities and the body and human health. You'll have normal microbiota everywhere in the body, from your eyebrows, from your nose, inside your gut as well, all the way through. Now, the distribution and composition of normal microbiota are determined by many factors, including the nutrients that are present in that area, physical and chemical factors, host defenses. Does the host get rid of those certain types of bacteria, or does it allow them to stay? And then mechanical factors as well. Microbial antagonism, or competitive exclusion, is a competition between microbes, which in the case of normal microbiota is a good thing. Our normal gut bacteria and bacteria in our body protects us, or protects their host, by competing for nutrients, preventing them from being readily available to harmful bacteria, and also producing substances that are harmful to invading microbes, and by affecting the pH and the level of oxygen that would otherwise harm bacteria that we would not want in those parts of our body. Symbiosis is the term used to describe the mutually beneficial relationship between normal microbiota and the hosts. There's commensalism, which means that only one organism benefits, while the other host or the other organism is not affected, negatively or positively. There's mutualism, which means that both organisms benefit in the end. But in the case of parasitism, only one organism will benefit to the detriment of the other organism. Some normal microbiota are opportunistic pathogens. For opportunistic pathogens, essentially, they see an opportunity and then cause a disease in the body. Say when you're taking medication and your gut flora gets diminished in concentration and something infectious can enter in at that point. There's a series of postulates called Koch's postulates, and these are from 1 to 4. The same pathogen must be present in every case of a particular disease. The pathogen must be isolated from the disease host and grown in pure culture, the pathogen from the pure culture must cause the disease when it's inoculated into a healthy, susceptible laboratory animal. And the pathogen must be isolated from the inoculated animal and must be shown to be the original organism. These are just a series of ways of showing to confirm if a certain disease is linked to a certain microorganism. Now, Koch's postulates, they're used to prove the cause of an infectious disease, because it's not normally good enough to just know what the symptoms of something are, but generally if you know the cause, you can treat it or prevent it. There are exceptions to Koch's postulates, though. Some pathogens cause several disease conditions, not just one in particular. And there are pathogens that cause disease sometimes only in humans, and therefore cannot be tested in lab animals. And then there are some microbes that have never been successfully cultured. Now, in the case of classifying infectious diseases, there are a few ways. One of them is by the symptoms, or the changes in typical body function that are felt by a patient as a result of a disease, such as is there an increase in temperature or a fever, or weakened energy. There are signs which are changes in the body that can be measured or observed as a result of a disease. And then there is a syndrome, which is a specific group of signs and symptoms that accompany a particular disease. In the case of communicable disease, that is a disease that is spread from one host to another. Now, a contagious disease would be diseases that are easily and rapidly spread from one host to another. And non-communicable disease is simply a disease that is not spread from one host to another. As far as measuring the occurrence of particular diseases, an incidence is simply the number of people who develop a disease during any particular time period, whereas prevalence is the number of people who develop a disease at a specific time, regardless of when it first appeared. The latter takes into account both old and new cases. For a sporadic disease, that simply is something that causes or only occurs occasionally but inconsistently. An endemic disease is a disease that constantly presents itself in a population. An epidemic disease would be a disease acquired by many people in a given area in a short amount of time. And a pandemic disease would be one that affects a worldwide population. When looking into the severity or duration of disease, one classification is that a disease may be acute. Simply, symptoms develop rapidly, but the disease lasts only a short amount of time. There are chronic diseases, though, where symptoms develop slowly. A subacute disease would be when the intermediate, it's a in the middle ground between something that's acute and something that's chronic. And a latent disease involves a causative agent that is inactive for a time, but under the right conditions becomes activated and produces symptoms of that disease. An important concept with disease prevention is herd immunity, which is simply the immunity when a majority of a population becomes immune to a particular disease, helping prevent the spread to other people in that population. When looking at the extent of host involvement, there is a local infection, where pathogens are limited to only a small or particular area of the body. For systemic infections, also called generalized infections, this is an infection throughout the body. And then focal infection is a systemic infection that began as a local infection, but eventually spread throughout the body. Sepsis would be a toxic inflammatory condition which arises from the spread of microbes, especially in the case of bacteria or their toxins, from a focus of infection. Bacteremia is when bacteria are in the blood that should not be there. Septicemia is the situation which is also called blood poisoning, which involves the growth of bacteria in the bloodstream. Toxemia involves toxins entering or proliferating in the bloodstream. Viremia is when there's a blood condition involving viruses. Primary infection is when there is an acute infection that causes the initial illness, whereas a secondary infection involves an opportunistic infection after a primary or predisposing infection, where, say, an initial disease or infection weakens a host, and then a secondary infection can then take hold. There are subclinical diseases in which no noticeable signs or symptoms are noticed or even any apparent infection, but the disease could possibly infect other people. Things called predisposing factors. They make the body more susceptible to a disease. Sometimes it may be based off of one's gender if they're more susceptible to one disease versus another. Sometimes it could involve inherited traits, which includes diseases like sickle cell anemia from the sickle cell gene. Sometimes climate and weather can make certain pathogens more proliferate better. And sometimes just being tired and not getting a good rest, or being older or younger, could make one more susceptible to certain conditions. Also, lifestyle, level of nutrition, and whether or not chemotherapy is being applied. Diseases develop in periods, one being the incubation period, which is the interval of time between the initial infection and when first signs and symptoms are observed. The prodromal period is a short period after incubation, early on when mild symptoms are appearing. The period of illness is when the disease is at its most worst or severe. The period of decline is when signs and symptoms subside, and then the period of convalescence is when the body returns to its pre-diseased state. Now, reservoirs of infection. These are just continuous sources of infection. Human reservoirs are a situation where humans can be carriers and may be inapparent infections, or they may have latent diseases within them that have not activated or become reactivated yet. There are animal reservoirs. Zoonoses are diseases transmitted from animals to humans. And then there are non-living reservoirs, such as pathogens that may be present in soil and water, and when ingested or entered the body, can begin to infect. Types of contact transmission, such as direct contact transmission, which requires close association between the infected and a susceptible host. There is indirect contact, where the spread occurs to a host by a non-living object called a fomite. And then droplet transmission, where transmission via airborne droplets within less than a meter of the initial release. As far as a vehicle or a pathway for transmission, transmission can be an inanimate reservoir, such as diseases that are within water, food, or in the air itself. Vectors, which are agents that carry a disease to a new host, or can be a source of a disease. They can include arthropods, especially those of fleas and ticks and mosquitoes, which transmit disease typically in two general methods. Mechanical transmission, where an arthropod carries the pathogen on its feet, or biological transmission, where a pathogen reproduces inside that vector and then is transmitted via bites or feces from the animal. Healthcare-associated infections, or HAIs. They're acquired while receiving treatment in a healthcare facility, also known as nosocomial infections, and will typically affect 1 in 25 hospital patients. About 2 million people per year get infected, resulting in 20,000 deaths. Now, when it comes to healthcare-associated infections, they can be caused from microorganisms in the hospital environment. They can affect because of the priorly weakened status of the host, or it could result from a chain of transmission from personnel in the hospital, from one person to the next. In a compromised host, the individual's resistance to an infection is impaired or weakened by disease, by therapies, or even burns. And the intersection of HAIs involves microorganisms in the healthcare-associated environment, the host already being weakened from some form, and some pathway to eventually reach that host. In order to control or limit these, it involves reducing the number of pathogens, reducing the number of pathogens that eventually reach someone being cared for, such as hand washing, disinfecting tubs used to bathe patients, cleaning instruments very meticulously and scrupulously, using disposable bandages and intubation, and performing infection control committees to help monitor and keep up healthy standards. For emerging infectious diseases, these diseases are new, increasing in incidence, or showing a potential to increase in the near future. Most are zoonotic, originating from some animal before coming to a human. They are typically of viral origin and are likely to be vector-borne, coming from some outside source into a person. There are contributing factors, such as genetic recombination, where bacteria or two different species of pathogens combine their infectious genetic material into a new strain. There is evolution of new strains where things naturally mutate with time, possibly becoming more infectious. The widespread use of antibiotics in situations where they're not warranted, as well as the use of pesticides, have resulted in pathogens that naturally mutate and survive, that naturally built up an immunity or mutated to have an immunity to antibiotics and pesticides, no longer have as much competition from the remaining microbes that were eliminated from antibiotics or pesticides. Therefore, those that do remain have no competition for nutrients and resources and can spread and grow quickly. And changes in weather patterns can also contribute to disease as well, such as with the hantavirus. There are situations where modern transportation causes a virus or some pathogen to easily or more easily spread from one area to the next, especially since the world is becoming more connected through airplanes, travel, ships, the variety. There are ecological disasters, tornadoes, tsunamis, and things like war or the expanding human settlement that can cause diseases to occur or become available to humans. Certain animal control measures that either have been laxed or not in the proper amount of control could result in certain diseases spreading more easily to people, such as in the case of Lyme disease, and in the case of public health failure, where certain facilities are not maintained properly, such as clean water infrastructure, which may result in diphtheria. Now, epidemiology is the study of where and when a particular disease occurs and how that disease is transmitted in those populations. Epidemiologists will study or perform work to determine the etiology of a disease, to identify other important factors that are involved in the spread of that disease, develop methods for controlling a disease or limit its spread, and assemble data and graphs to outline incidence of a disease. Now, there are many big names in the field of epidemiology, such as from the 1900s, John Snow, who mapped the occurrence of cholera in London, Ignaz Semmelweis, who showed that hand washing decreased the incidence of puerperal sepsis, Florence Nightingale, who showed that improved sanitation decreased the incidence of epidemic typhus. And, well, certain graphs will show that certain diseases either have decreased in prevalence with time, such as the introduction of the polio vaccine has had on polio incidences, or certain graphs will show how certain diseases are becoming more prevalent with time, such as how Lyme disease seems to have grown with time from 1999 to 2013. And how sometimes graphs can show how there may be certain localized months or seasons where a particular disease is more common, such as during the summer months when ticks become more active, and where ticks can be a vector of Lyme disease, an agent that spreads it. In the case of improved healthcare and medical know-how, tuberculosis cases have drastically dropped well over the past few decades. Now, types of epidemiology. There is descriptive epidemiology, which is a collection of relevant data and then the analysis of it. For example, what was performed by Snow. There is analytical epidemiology, which analyzes a particular disease to determine its most likely cause, such as in the case of Nightingale's research. In the case of Semmelweis, they performed experimental epidemiology, which involves a hypothesis or forming a hypothesis and then performing controlled experiments to test that hypothesis of a disease's spread. Centers for Disease and Control and Prevention, or the CDC, is a primary source in the US that collects and analyzes epidemiological information. It publishes an article called Morbidity and Mortality Weekly Report, where the two terms morbidity means the incidence of a specific notifiable disease, and mortality is the death from that disease. Other things involved in the CDC involve notifiable infectious diseases, which are diseases in which physicians are required to report the occurrence of. There are morbidity rate, which is the number of people affected in relation to the total population in a given time period. And then mortality rate, which is the number of deaths from a disease in relation to the population in a given particular time. And such notifiable diseases include a wide variety, including many major ones, from anthrax to rabies, even leprosy, among others. As far as clinical focus, in the case of healthcare-associated infections, blood cultures grown on mannitol salt agar, coagulus positive and gram-positive cocci, methicillin-resistant Staphylococcus aureus, which is a strain where 92% of healthcare strains, that is the cause of 92% of healthcare strains. Or in case of strain USA 300, which is the result of 89% of community-acquired strains in healthcare-associated infections. And that concludes this lesson in Chapter 14. Well, this concludes this lesson in Chapter 14. And anyways, peace out, everyone.